lot of times they will ask questions that talk about radio labeled things, right? So scientists are able to put radioactive labels on molecules. Usually they're like phosphorus or sulfur, radioactively labeled atoms that allow you to track where molecules are going. That's all that's happening. So in an experiment, researchers provided a radio-labeled amino acid. In your mind, where are you classifying amino acids? You should be thinking proteins right away because amino acids are part of proteins. Okay. After an hour, the researchers determined the amount of radio-labeled amino acid that was in each of the several subcellular components, in other words, each organelle, right? So try to translate it into English for yourself. The results of the experiment are here. So in the nucleus, you had 2.1 of these. The mitochondria, you had 2.7. So you have the most located in the mitochondria and the least out in the cytoplasm, okay? Which of the following conclusions about the amino acid is best supported? So you're thinking, well, if there's mostly in the mitochondria, you should be thinking about that. It was mostly incorporated into proteins that regulate and manage metabolic reactions. So let's think about that for a minute. Why would that go best with, mitochondria or nucleus? What's a metabolic reaction?
A reaction that produces waste.
Yeah, well, it might be very well. A metabolic reaction is just any of the chemical reactions that are digesting or biosynthesizing molecules. So metabolism is, we're going to get into this in the next unit. Metabolism is just all the chemical reactions happening in an organism, really. That's all it is. And some of those reactions might be digestion of food. Some of them might be biosynthesis, putting things together, like building proteins, making fats. Those are all metabolic reactions. That's all that means. So in terms of where are the most metabolic reactions, they would be in the mitochondria probably because there's a lot of breakdown and digestion happening in the mitochondria as they take the sugars apart and use the energy to make ATP. Any questions about that one? Okay. Right, so A, if the nucleus had been really high, then you would have said A was the answer because they're talking about something that happens in the nucleus, right? C, it was mostly incorporated into lipids that help separate cells from their surrounding environment. Maybe that would have been if the ER had been the highest number, then you might have picked that one, okay? Or it was mostly incorporated into carbohydrates that form protective structures outside of the cells. Not sure where that one would have gone. Maybe in the cytosol or something like that. That's kind of neither here nor there. It's more of a distractor. Okay, you guys with me? Okay. All right. Number 5, researchers conducted an experiment. A lot of times they're going to give you data and then say, figure this out, right? To investigate the effects of valinomycin treatment. And you don't have to be able to pronounce it. You just need to be able to make a conclusion about it, right? On skeletal muscle cells. Valinomycin is a naturally occurring substance that can be used as a drug. The results of the experiment are presented. So there's time. This is going through time. The cells that didn't get the drug went from 1 to 7.7 in rate of ATP production. The cells that did get the drug went from 0.3 to 2.7 in ATP production. So without looking at the answers, is valinomycin helping to increase ATP production? No, it is not. Okay, so with that in your mind, now look at the answers. And the actual answer is this one. The valinomycin treatment caused a decrease in the activity of the mitochondria. Yes? Okay, and again, they're connecting ATP production with mitochondria, which you should be doing, right? Because ATP is energy, and the mitochondria is all about making that energy. All right. Any questions on that one? Did anybody already do all these and just wants me to go over 1 or 2 of them? Too much to hope for?
Did you go over 13?
13. And I have to tell you that, exactly gone through every single one of these myself, so I'm right with you. Okay, the intravenous IV therapy. That's when they hook your blood up to, you know, to an IV sac of saline. Is used for fluid replacement in instances of dehydration in humans. One type of IV fluid is essentially a salt water solution. To determine the best concentration for therapy in people, a team of students is researching the effects of solutions of different salt concentrations on red blood cells. The following observations were made from 3 different red blood cell samples viewed under the microscope. So what should a red blood cell look like normally? Should look like the middle one, right? That's a healthy red blood cell. This looks all bloated up with water. We just got through doing all of this, right? With the essay that you just wrote. And this one looks all shriveled up. What is a red blood cell's favorite tonicity? Isotonic. It likes to have the same concentration around it as inside it so that it doesn't swell up or shrink, right? So this is the happy cell here. So knowing that, the team wants to extend the research project. What should the team of students do next to obtain data that are most conclusive? So let's look at the choices. Repeat the process with other salt concentrations. Okay. Develop a model to explain why the cells react differently to different salt concentrations. Repeat the process using red blood cells from other animals. Develop an experimental procedure that uses a stain that makes the organelles of red blood cells more visible. What do you think? What did you think?
I thought it was C because just the first sentence of the question mentioned other animals. So if they want to be sure, then maybe they could test it in animals. But I was also going between that and B.
I was looking at D myself, meaning if they want something more conclusive, they would want to know not just about the overall shape of the cell, but they might want to know what's going on with the organelles inside it. And I'm saying that without knowing what the answer is. So, other animals. That's right. Well, I'd say that's actually a really difficult question. You know what the best answer to that is? I am guessing that it's C or D, but let's see.
They're asking for the different cell concentrations.
Look at that. I don't like that question.
Aren't there like a bunch of things that you can do to make something more conclusive? Like it's so broad.
That is awfully broad. I don't like that question. I'm not going to ask you that. What should the team of students do to obtain data that are more, that is really vague. Okay, have we established that we don't like this question and that Mrs. Murdoch is not going to ask it? Excellent. But you did learn something from it anyway because we talked about isotonicity in animals, right? 16? Here's 16, okay. So, okay, we're about lysosomes digest food particles brought into a cell by endocytosis. What's endocytosis? Yeah, so it could be phagocytosis or pinocytosis, but one way or another, the whole membrane is moving to pull things in, right? Okay. After vesicle-containing food particles fuses with a lysosome, hydrogen ions are transported into the lysosome from the cytosol. This significantly lowers the pH of the lysosome relative to the cytosol and activates the enzymes that digest particles. Which of the following best predicts what will happen to the lysosomal enzymes if the proteins that transport hydrogen ions from the cytosol into the lysosome are damaged? Okay. A, the lysosomal enzymes will not become active since there will be no active transport of hydrogen ions. The lysosomal enzymes will not become active since hydrogen ions will diffuse out of the lysosome. The lysosomal enzymes will become active since facilitated diffusion will move hydrogens into the lysosome. The lysosomal enzymes will become active since passive diffusion will move hydrogen ions into the lysosome. Okay. if the transporters that are supposed to bring them in are damaged. Well, let's see. Okay. Did you guess that one? The lysosomal enzymes will not be active since there will be no active transport. Well, I mean,
It says
that's kind of a gimme, isn't it?
the only one is like the only part that I got it was because it says activates the enzymes that digest.
Right. Okay. So use the question to answer the question?
Yep.
Yeah. Okay. So, right, I mean, if there's no transporter, all the rest of them are talking about the ions are moving. How are they moving if there's no transporter for them? So that doesn't make sense. It's almost so obvious that you're afraid to pick it. Sometimes it's like that. Okay, how are you doing? Yeah. 23 and 27. Okay. Some viral infections can lead to the rupture of the lysosomal membrane. Which prediction of the effect of this disruption of cellular compartmentalization is most likely correct? And the answer is C, hydrolytic enzymes will be released, which will cause death. Okay. Okay, so a viral infection that causes a lysosome membrane to rupture. A lysosome's other name is called suicide sac. So if you have a lysosome that breaks open, their usual purpose is to go through programmed cell death. That's what a lysosome is sometimes for, right? So if it breaks open, it can cause programmed cell death. Does that help? Okay. Yes. Which one? 27?
Wait, Miss Murdoch, what do lysosomes do normally?
Lysosomes, what they do normally, like for example, they contain digestive enzymes, right?
Okay.
So for a single-celled amoeba, a lysosome is like a stomach. An amoeba will eat food, and the lysosome will fuse with it, and the lysosome breaks down the food for the lysosome like a stomach would, and then releases the food so the amoeba can use it. In an animal cell, lysosomes will recycle anything in the cell that is risky, right? There's something called apoptosis, which is programmed cell death, and we're not quite there yet, but if the cell is determined to have DNA that's damaged in it, there are ways for the cell to do that. When it's replicating its DNA, it finds a mistake and it doesn't get corrected, and the cell starts to show signs of becoming cancerous. Lysosomes will be instigated to break open and kill the cell before it can cause a tumor in the animal. This is a good thing. Programmed cell death is a good thing when you have a cell that has damaged DNA. So, I mean, there's other uses for lysosomes. Animals have a lot of them. It depends on what kind of cell it is. But that's its basic use. Does that help? Mm-hmm. Okay. A euglenid is a protistan. I love euglenids. They have these funny tails. Okay. So it's a protistan that has, this would be like looking at pond water, right? And you have this cell that has a flagella coming out of it, and it's really fun, okay? It's a single-celled eukaryote that live in aquatic environments. The chloroplasts found inside, here they are here, are enveloped by 3 membranes. The inner membrane of euglenid chloroplast resembles the thylakoid membrane. Okay, which of the following claims about the origin of euglenid chloroplast is best supported by the 3-membrane structure of the envelope? So we're going towards endosymbiosis here, right? Okay. It originated from the spontaneous assembly of organic molecules into a lipid bilayer inside a free-living prokaryote. It originated from the fusion of plasma membranes of 2 different free-living photosynthetic prokaryotes. It originated from incorporation of a photosynthetic prokaryote into a eukaryotic cell by a single endosymbiotic event. It originated from a photosynthetic prokaryote into a eukaryotic cell by 2 endosymbiotic events. I know which one I'm picking. Think of a little sort of prokaryote that knows how to do photosynthesis, right? Look at that picture up there on the wall. That's endosymbiosis. It's basically phagocytosis, except that the cell that got engulfed never got digested. Instead, it becomes a permanent resident, right? So I think what we're looking at here is a little a bacteria that can do photosynthesis, and there are some that can do that got engulfed, not just once, but twice, so that it gave it a three membranes around it in the end. I hope I'm not wrong about this, but I think this is right. Yes. All right. Okay. So, endosymbiosis, some more of the questions tend to be about what the evidence for it is. So the first chloroplast that was ever engulfed, the reason why we now have chloroplasts that we think got there by endosymbiotic theory 2 billion years ago is because modern chloroplasts in plant cells have their own DNA. They have their own DNA, they have their own ribosomes, and they replicate just like bacteria, as if they were still independent livers. Same with our mitochondria in our own bodies. Mitochondria got engulfed just like that and became the first mitochondria in the eukaryotes. We think this is true because mitochondria also have their own DNA, they have their own ribosomes, and they can divide just like bacteria do, as if they were still independent livers. But what this question is going with is, now we're also kind of trying to explain why you have some organelles like the nucleus right now that doesn't have one membrane, it actually has 2 around it. It's got a double membrane. And that's probably because when it was engulfed, it kept the phagocytic, I don't know if you understand what I'm saying. When phagocytosis occurs, right? Here, let me draw it. When phagocytosis occurs, when one cell eats another cell, right? So you have, here's a cell, here's another cell that's going to eat it. And then it goes in, right? It keeps this membrane around it. So now this is a double-celled, double-membraned organelle, right? So ostensibly, what could have happened in that question is you could have had a chloroplast engulfed by something not much bigger than it, right? So then you have this. And then it got eaten again, giving you a triple membrane system. So endosymbiosis isn't about just single organelles like chloroplasts and mitochondria getting eaten and staying alive and becoming symbiotic. It's also about creating these multiple membrane systems. This is probably the way we got our ER and the way we got our nucleus with its double membrane and the way we got a lot of the things that are inside some of our cells that have multiple membranes in them. Good? Okay. I have enough energy left for a couple more questions. What would you like to do? Yeah.
Could you do question 20?
20?
Yeah.
Okay, and then Sophie, you can ask one, okay? A magnesium sulfate solution, sorry, I have the answer off, taken orally can cause a net movement of water into the large intestine, which results from water molecules diffusing through aquaporins embedded in the cells of the intestinal lining. By which of the following mechanisms do water molecules likely move into the large intestine? Okay. Magnesium sulfate by passive transport from an area of low osmolarity to an area of high osmolarity? Okay, osmolarity, you don't know that word, do you? And it's confusing, and it's actually very simple, okay? Osmolarity Osmolarity is another word. It's pretty much the same as concentration. So it's just how much solutes are in there. That's all it means. That's all it means, okay? So if we go back to the question, they're going to try to confuse you with these big words, and they're just talking about stuff that you really do know, okay? So replace osmolarity with just the word concentration, solute concentration, okay? By passive transport from an area of low solute concentration to an area of high solute concentration. So would the water move passively like that?
Yep.
Yeah. If you did your paired essay well, then you know that can work. Let's make sure that you understand this. Right? So high solute concentration, right? So from an area where, I don't know, there's a membrane here, okay, here's another area there, and we have from an area of low solute, right? So let's say that this is salt, there's your membrane, and there's a lot more over here. Okay? Okay. The water will move from where it's high, right? So maybe the osmolarity is 0.2 over here and 0.8 over there. So there's less salt here and more here. The water will move that way, won't it? Yes. Okay. I hope I got him right. Yeah. Yeah. Okay. All right. So osmolarity just means solute concentration. That's all it means. Okay. You'll probably see that word on the AP exam. You might see it on mine. Does that help?
Yeah, I just got confused because I thought it was talking about the water concentration.
It sounds like it's talking about water concentration, doesn't it? It's actually talking about the solute concentration. Yeah. Good. Okay. Sophie, what was yours?
Number 15.
Number 15. Okay, so researchers investigate the transport of a certain protein into cells by endocytosis. In an experiment, the researchers incubate the cells in the presence of the protein, and measure the amount of the protein that is absorbed into the cells over a 5-minute period. Based on their observations, what should the researchers do to further clarify how the availability of the protein outside the cells affects the rate of endocytosis of the protein? Okay, I don't know why you'd incubate it in the absence of the protein. That wouldn't do any good, would it? Incubate the cells in the presence of several different proteins. That muddies up your IV, right? Incubate the cells in the presence of several different concentrations of the protein, maybe. Incubate the cells in the presence of the protein for several different lengths of time. How the availability of protein out the cell? Well, then it would be C, wouldn't it? Yes. Okay, so all I did was use their wording, right? So what should the researchers do to clarify how the availability of the protein outside the cell affects the rate of endocytosis? Availability, in other words, concentration of protein, right? So incubate the cells in several different concentrations of protein, and that answers the IV they're looking for. Okay, how are you guys doing? Yeah, I mean, you definitely should try these difficult questions. This is the way to go. And go back and go over your notes, go over your guided readings, go over some of the YouTubes. Can you redo the egg puzzles? I don't even know. But you can, I mean, they're all YouTubes that I found online. You can go get them, right? You can do that, if that helps. But that's exactly what you need to do, probably, right? That's good. You should do that. Okay. All right, that is as much as my voice can't do anymore.